Hello everyone, welcome to Earth Archives. Please take a moment to subscribe, like, comment, share, and don't forget to hit the bell icon. Thank you. Today's topic is the Immortal HeLa cells. Immortality has been a goal for humanity throughout history, from emperors and kings to alchemists and magicians, all seeking this ultimate prize. But has anyone truly succeeded? Aside from legends, no one has actually achieved it, right? But today, what I want to talk about is not humans, but the history of a human cell that gained immortality. It all started with a 2010 newspaper headline. Cancer cells killed Henrietta Lacks, 1920 to 1951, but also gave her immortality. Strictly speaking, it wasn't Lacks herself who achieved immortality, but rather her cancer cells. Henrietta Lacks lived in an era when cancer was feared by all, and medical treatments for the disease were limited. Cancer was often a taboo subject, with families keeping quiet about it. Medical researchers were struggling to find a cure for cancer, with the biggest challenge being the inability to grow cells outside of the human body and conduct experiments. Through a dramatic process, scientific research finally overcame this obstacle, ushering in a new era of cell research, the era of HeLa cells. Scientist George Gay successfully kept Henrietta Lacks's cervical cancer cells alive in a petri dish and shared these cells and the cultivation method with researchers worldwide. These cells were named HeLa cells, derived from the first two letters of Henrietta Lacks's first and last names. Prior to this, human cells could only survive for a few days in culture, but HeLa cells thrived in artificial environments, reproducing every 20 hours and continuing indefinitely as long as the conditions were right. Because of their incredible vitality, by the 1960s, scientists found that HeLa cells could even contaminate other cell cultures in the lab. According to renowned obstetrician-gynecologist Howard Jones, 1910, the tumor he observed in Lacks was unlike anything he had ever seen before, purple in color, prone to bleeding upon touch and growing rapidly. I had never seen such a cancerous tumor before, and I've never seen one like it since, he said, describing it as an unprecedented case. Once HeLa cells were successfully cultured, a wide range of scientific research experiments began using these cells. Almost anything you can imagine, even some near science fiction scenarios, was tested on HeLa cells, including adding various toxins to study their reactions and even sending the cells into space to observe the effects of zero gravity on human cells. HeLa cells were also essential in the development of the polio vaccine. Despite their significance in medical research, HeLa cells were not widely known within the medical community. They were often vaguely described, and it wasn't until a specific event that the full truth about them was revealed. To understand the impact of these cells, we must first learn about Henrietta Lacks herself. She was a descendant of white landowners and enslaved black people, raised by her grandfather in a slave quarters. Before giving birth to her fifth child, she told her family that she had something inside her, but both her family and doctors dismissed her claims. In early 1951, Henrietta Lacks was diagnosed with cervical cancer at Johns Hopkins Hospital and underwent radiation therapy with radium. By early October of the same year, her cancer had advanced, and like many cancer patients of that time, she succumbed to the disease after extreme suffering. Lacks chose to seek treatment at Johns Hopkins because the hospital provided free medical care to the poor. However, due to her family's poverty, there was no money for a tombstone after her burial, and her 5 young children were left to face life's hardships without their mother. The lack of financial resources compounded their difficulties, illustrating how poverty has been one of humanity's greatest challenges throughout history. The issue lies in the fact that, before her death, researcher George Gay at the hospital collected cancer cells from Lacks without her knowledge for experimentation. During the 1950s, Johns Hopkins Hospital offered free medical care to many poor patients, many of whom were black. It was common practice at the time for doctors and researchers to take tissues or cells from these individuals without obtaining informed consent. However, the story didn't end there. Lacks had a tumor the size of a baseball that nearly replaced her kidney. The cancer cells were unusually active, and Gay's laboratory quickly discovered that while other cell cultures would die within days, cells taken from Lacks had an exceptionally long lifespan. Gay's assistant, Mary, also observed that the cells grew rapidly, doubling in number almost every day, capable of dividing endlessly. The speed at which these cells grew was truly remarkable and alarming. Mary even transferred the cells to new petri dishes, and soon they would fill them up again. For convenience, she labeled the petri dishes with the initials of Henrietta Lacks, which led to the cells being named HeLa cells. George Gay used the HeLa cells to establish a cellular system, sharing his discovery with other research institutions. Even when Gay himself was diagnosed with cancer, he attempted to create a second generation of HeLa cells from his own cancerous cells, but unfortunately, his attempt failed. To this day, HeLa cells remain the epitome of immortality in cellular biology. It's important to understand that the limit of cell division is typically 56 times, known as the Hayflick limit. Each division consumes the DNA's telomeres, and once these are exhausted, the cell dies. This process is part of why humans age and die. However, HeLa cells and cancer cells in general are different. During division, they can use an enzyme called telomerase to replenish the telomeres, which allows them to bypass the usual limit on cell division. HeLa cells' immortality made them the ideal experimental subject for countless studies. They became essential in virus research and many other scientific applications. One of the first breakthroughs using HeLa cells was in the development and testing of the polio vaccine. In 1954, Dr. Jonas Salk was finally able to develop the vaccine with the help of HeLa cells. This was one of the greatest medical victories of the 20th century, saving countless children from the deadly disease. HeLa cells also led to the creation of the world's first cell factory, the Tuskegee Institute's Cell Sales Center. This facility could produce up to 20,000 vials of HeLa cells per week, equating to 6 billion cells, an astonishing number. Over the decades, HeLa cells have been crucial in AIDS research, drug development, and the rise of virology. In 1965, Henry Harris and John Watkins successfully combined HeLa cells with mouse embryo cells to carry out experiments on gene mapping, which paved the way for the Human Genome Project. To this day, HeLa cells have divided and grown into more than 18,000 generations, a number roughly equivalent to the 450,000 years it took for humans to evolve from early primates to Homo sapiens. Research into immortality using HeLa cells has been ongoing, but due to the sensitive nature of the subject, mainstream media has often refrained from extensive coverage. In modern times, HeLa cells have become a major focus of medical research, generating substantial profits for medical institutions. However, Henrietta Lacks' family remains in poverty. As her daughter, Deborah Lacks, said, "I've always found it strange. If my mother's cells contributed so much to medicine, why is it that her family couldn't afford to see a doctor? It just doesn't make sense. Many people became wealthy because of my mother, and we didn't even know they had taken her cells. Now we don't have a dime. I used to get so angry thinking about it. I was really angry, so angry it made me sick, and I had to take medication for it." Over 50 years after Henrietta Lacks' death, the scientific community finally began to address the issue. Johns Hopkins University and the Lacks family formed a committee to determine how HeLa cells should be used and by whom. When researchers first invited Deborah Lacks to visit the laboratory at Johns Hopkins and see the HeLa cells, she stood in front of the frozen vials and said, "I can't believe this is my mother." She blew on the test tubes and said, you're cold, you're famous. We can make an assumption. If Henrietta Lacks had been a middle-class white woman, the exciting scientific developments driven by her cells might never have happened. She would likely have been treated in a different kind of hospital room, perhaps even receiving care at home. For the medical personnel and researchers of the 1950s, taking tissue or cells from a patient's body seemed like a natural thing to do, to the point that they didn't consider the ethical implications. According to American law at the time, once a patient underwent medical procedures or surgery, any leftover tissues or body parts were considered the property of the doctor or the hospital, not the patient. Regardless of the ethical issues, we cannot deny that the availability of numerous medications and treatments for diseases today owes much to the Immortal HeLa cells. Without their incredible ability to grow, even the development of the polio vaccine might have been delayed.